cell cycle, how I talked about how most of the cells in our organs are not cycling. The only times that our cells actually are into the cell cycle is when there's been an injury, where cells have to be replaced, or if cells have died, they have to be replaced. But for the most part, our cells in our bodies stay in G0, and that's a good thing. That's a really good thing. The reason why you want your cells to stay in G0 is because of our telomere sequences. You're looking at a micrograph of chromosomes, and there's a structure at the end of all of our chromosomes. It's called a telomere. And telomeres have an interesting sequence. This sequence, TTA-GGG, is repeated out at the ends of our chromosomes about 1,000 to 2,000 times. What's interesting, and the probe that picks up the telomere sequences is shown here in the bright dots. Telomeres, not only are this sequence repeated time and time again, but telomeres also have associated with them protein complexes. So the ends of our chromosomes aren't just waving free out into the nucleus. The ends of our chromosomes are associated with protein complexes, and all of the proteins associated with their telomeres have not yet been identified, certainly not for human telomeres. But the ends of our chromosomes are the repeating sequence associated with protein complexes, and this protects the ends of our chromosomes. But when a cell goes into the cell cycle and DNA has to be replicated, what's going to happen? What's going to happen is the RNA primer. Remember, during RNA synthesis, an RNA primer has to sit down, and then you have synthesis of the DNA in a 5' and 3' direction away from the RNA primer. The problem is out of the ends of our chromosomes, that RNA primer actually sits down about 50 to 100 nucleotides into the end of our chromosome. Not right at the end, but about 50 to 100 nucleotides in. So all of the DNA, these 50 to 100 nucleotides that are hanging out, that the primer hasn't attached to, those are going to be lost. So you lose about 50 to 100 nucleotides on the ends of your DNA with every cell cycle. Okay? That's not a good thing, because what happens when the telomere gets about 3,000 bases in length, our cells will go into crisis and they'll die. All right? So you want to have your cells in G0. The fewer the cell replications you have, the better you are. Okay? And that's called the end replication problem. How telomeres lose 50 to 100 nucleotides with every S phase. It's called the end replication problem. It has to do with where that RNA primer attached to the end of the chromosome. And again, these RNA primers will only attach about 50 to 100 nucleotides in. Therefore, these are going to be lost every time. Okay? All right. Does anybody have a question, or can I repeat anything? Question? The end replication problem. Is any of this in your book? Absolutely not. Okay. All right. Okay, we're going to move on. All right. So one of the problems with human aging is you have a slowdown in metabolism. You've all heard this, right? As you age, the metabolism slows down. What does that mean? What does that really mean? Well, I'm here to tell you. Okay, so what happens over time? time, when you're making ATP, electrons are delivered to the electron transport chain and carried along, and electrons eventually are delivered to oxygen. That's the electronegative atom that pulls electrons down the electron transport chain. About 15% of the oxygens that are being reduced as a result of electrons coming down the electron transport chain, about 15% of oxygens are insufficiently reduced. And those insufficiently reduced oxygens, as indicated here, become what are called superoxide free radicals. These are very, very reactive species. They will actually tear through the cell membrane. They'll tear through the chromosomes. They'll tear through proteins, looking for something to finish reacting with so that this superoxide free radical will lose its excess energy and become fully occupied with electrons. So in order to do that, it is tearing through the phospholipid bilayers of the mitochondria. It's tearing through the proteins of the cell. It's damaging the chromosomes specifically in the mitochondria. So mitochondria, over time, because of the accumulated damage from superoxide free radicals, the superoxide free radicals are over time damaging our mitochondria. First off, those superoxide free radicals are ripping through the mitochondrial DNA, causing mutations in the mitochondrial DNA, ripping through the proteins of the mitochondria and the proteins of the electron transport chain, the phospholipid bilayer of the mitochondria. So it's a terrible fact of life that as we make ATP, there are oxygen atoms that are not being fully reduced. Those oxygen atoms become superoxide free radicals, and they can do serious damage to the mitochondria. So over the course of 30 or 40 years, when all of a sudden, you at age 20 can eat what you want, exercise as little as you want, and you still look fabulous. You look a lot more fabulous than you think you do, believe me. Because by the time you're 40 or 50, what's going to happen is you're exercising more than you ever did. You're eating less than you ever did, and you weigh 1/3 as much as you did. You weigh 1/3 more than you did back in your 20s. So why is that? Why is that? That's because over time, what has happened is your mitochondria have taken so much damage over time that your metabolism has slowed down, pure and simple. Okay? That's one of the reasons why your metabolism slows down, is that over time, your mitochondrial DNA that are encoding all of these critical proteins that are part of the electron transport chain, part of the ATP synthesis pathway, all of that is getting slower. There's the genes that encode these critical proteins are starting to sustain mutations. And if the mutation doesn't totally stop the protein, it can certainly affect its efficiency in action. I mean, it's not all a total loss, because we do have some protective mechanisms. The protective mechanism primarily, when superoxide free radical is generated, we have an enzyme called superoxide dismutase that's going to actually take a couple of these oxygen-free radicals, combine them with the very abundant hydrogen ions that are present in the mitochondria, and form hydrogen peroxide. Hydrogen peroxide isn't the end of the story, because hydrogen peroxide is toxic in itself to the cell. So we have another enzyme called glutathione peroxidase that will convert hydrogen peroxide to water. And that's why we can live till we're 81. If we didn't have a good functioning superoxide dismutase or glutathione peroxidase, you wouldn't even live that long, because this could be a very, very damaging process. Okay? Does anybody have a question? Can I repeat anything? Anybody aging here? You feel the effects of this? Yes. Okay. That's the sad thing. We really don't. All of a sudden, it's just, like I said, you start to gain weight. You don't have as much energy. It's just the natural process of aging, and now you know why. So one of the things, one of the experiments that actually gives credence to this pathway I just talked about and this process that I talked about, here's an experiment that actually shows what happens. And they use mice as their model system. And mitochondrial DNA polymerase. Mitochondrial DNA polymerase, that's actually involved in replicating our mitochondrial DNA, and many of the genes that encode the proteins involved in ATP synthesis, these are present on the mitochondrial genomes. And again, these things are sustained mutations. And in this experiment, they had a mitochondrial DNA polymerase that was defective. It was defective. And so take a look at the premature aging that occurred in mice that had the mutant or defective mitochondrial polymerase compared to a normal mouse. And here you're just looking at a skin section. Just looking at the skin. And this is normal cross-section of normal mouse skin. What you have is the stroma and the basement membrane, and then you actually have the keratinocytes that are dividing down along the basement membrane, and then they work their way up and die and flatten out just to become a keratin layer. But underneath, in that stroma, a normal mouse, isn't he nice? Nice round, white laboratory mouse living in what you call leisure world conditions. He has a nice fat layer right underneath his skin. In contrast, the mouse with the defective mitochondrial DNA polymerase, look at this poor thing. It has a serious hunchback. This is called a kyphosis. So the spine has become deformed. There's no fat underlying the skin itself. That stroma does not have that nice fat layer. Instead, this mouse is skinny, has a deformed spine, and this mouse is going to die by the time it's a year old. Whereas the normal mouse is going to live at least 2 to 2.5 years. Okay? Is that clear to everybody? Okay, so what this does, this is the experiment that actually talks about what would happen. What happens when the mitochondrial DNA is not properly replicated and repaired because the gene that encodes this particular polymerase has sustained mutation as a result of superoxide free radicals. Okay? All right. And so this is what you call an aging model. Not unlike the Hutchinson-Gilford patients that I showed you earlier. Okay. All right, now I want to spend a little time on the insulin pathway. It's known that people who have what we call restricted diets tend to live longer. People who have restricted diets tend to live longer. And I'm not talking about malnutrition. I'm not talking about trying to scrape by on 1,200 calories a day. That's dieting. That's hardcore dieting. I'm talking about the average caloric intake for maintaining normal body weight. For men, it's around a little over 2,000 calories a day. For women, it's a little bit lower. Hard to maintain in the world we live in where everything just looks so good and it's so abundant, right? I don't know how many times I see 20-year-old students sitting around, each with 2 pieces of pizza, plus a platter of fries for the table. Like I said, you can do it when you're 20. You're not going to find somebody who's going to do anything like that. That's because, like I said, our metabolism has slowed down, but also there seems to be an involvement with the insulin pathway. And models for aging include Drosophila, or fruit flies, mice, like I just showed you, and also C. elegans. These are little garden worms, little round worms, but they're a wonderful genetics model. And it was found that if you give plenty of calories to Drosophila, to mice, and to C. elegans, what happens is you activate the insulin growth factor receptor and the insulin pathway. The insulin pathway and the insulin-like growth factor receptor is called that in mammals. Its equivalent is INR in Drosophila and GAF2 in C. elegans. So we'll focus on this. Insulin-like growth factor receptor, when it binds to insulin, will activate protein kinase B. Protein kinase B, what it does is it inhibits the action of the FOXO transcription factor. The FOXO transcription factor activates genes that encode proteins that protect against reactive oxygen species. What would that be? Superoxide dismutase, right? And glutathione peroxidase. So the genes that actually protect us against reactive oxygen species, those are activated by the transcription factor FOXO. And the insulin-like growth factor pathway actually inhibits this. What FOXO also will do is it will activate genes that encode DNA repair enzymes. And so the FOXO transcription factor in mammals and its equivalent in C. elegans as well as Drosophila, what these transcription factors do is by protecting the genomes in our mitochondria and in our cells, it increases cellular lifespan and therefore increases overall longevity of the organism. And that's the actual basis for how, if you restrict your diet, you can extend your life, is through this mechanism right here. Actually, a very good friend of mine actually works on aging in Drosophila. And I remember he was so proud because his Drosophila in his lab, he was able to actually restrict their diet. And instead of living for 3 months, he could actually get them to live for 4 months. And he was so excited and proud of that because he was working on this pathway that he told his mother-in-law. And his mother-in-law just looked at him like he was crazy. He said, "Bob, we don't want flies to live longer." So anyway, you got to be careful what you talk about with your family. You know, they just don't understand necessarily your research. But nonetheless, this is one of the reasons why if you watch your diet and you limit your diet, you limit this pathway. So you're not going to have 2 pieces of pizza and a platter of fries for the table, if you think about this, because what's going to happen when you do that, you're restricting the action of your transcription factors that activate expression of DNA repair enzymes. Okay? Does this make sense to everybody? So what is the primary way that you can actually start to stop your aging process. Would you like to stop aging now? I mean, you might want to think about that. And so the way you do that, the primary way you do that is exercise. The more you move, keep your muscles toned, keep your blood moving. Don't let your fluids down in your tissues start to accumulate. Keep moving. Exercise is actually one of the most important things you can do. At least 30 minutes of brisk walking a day. And I can tell you at my age, that doesn't do it. I beg to differ. Have you been up to my office? Those stairs? I do that 5 times a day. Oh my God. Anyway, another thing you can do, and did I talk to you about this when I was talking about cancer? If you take a low dose aspirin every day, 81 milligrams, that's baby aspirin. That's not a regular aspirin, but a baby aspirin. You decrease your chance of developing cancer by upwards of 25%. And that's lung cancer, breast cancer, colorectal cancer. That's because low-dose aspirin is an anti-inflammatory, and it's the inflammatory pathway that allows for some tumors to develop. So take a low-dose aspirin. That'll help to protect you against cancer. Maintain your calcium levels, because that's critical for maintaining muscle and bone mass. Once you start losing muscle and bone mass, you can't exercise very effectively, and you start to age as a result. Then, acetyl-L-carnitine, right from the aging expert himself, acetyl-L-carnitine. It actually imports fats into your mitochondria and contributes to cellular energy. Alpha-lipoic acid, also, again, from my aging expert buddy. Mitochondrial specific antioxidant. It scavenges those reactive oxygen species that we talked about, so that way you can help to preserve your mitochondrial DNA and therefore your mitochondrial proteins that are so critical for allowing for ATP synthesis. Those are going to be able to function with full efficiency. Okay? Does anybody have any questions at all? The question is, what foods contain a lot of this? You know, that question I don't know. I just take them as we are. Okay, it was fun. Fun semester, everybody.